by Atta, who is an associate professor in the Department of Microbiology in Ahmadu Bello University, Zaria, Nigeria. Her area of specialization is environmental microbiology, with major research areas in petroleum microbiology, bioremediation, ecology and biodiversity, biofuels, and WASH, which I think it stands for water, sanitation, and hygiene. Her career as an academic spans a period of 18 years. She was a Fulbright visiting scholar in the year 2021 for postdoctoral research at Rutgers University in the USA. She was a beneficiary of the Petroleum Technology Development Fund postgraduate scholarship for her PhD research. She has authored several peer-reviewed journal articles, contributed to book publications, and is also an associate editor and book reviewer for the Fulbright Chronicles. Dr. Atta serves as a scoping expert and reviewer with the Intergovernmental Society, with the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services, IPBES. She is passionate about science communication and is a strong advocate of STEM education and environmental pollution issues. She is currently the institutional coordinator of the Organization for Women in Science for Developing World, OWSD, at Ahmadu Bello University branch. Dr. Atta is a member of the Nigerian Society for Microbiology, American Society for Microbiology, Society for General Microbiology, Society of Environmental Toxicology and Chemistry, SETAC, and many other professional bodies. I believe that her biography is longer than this, but this is what I have as a summary. So you're highly welcome, our distinguished speaker, Dr. Habiba Atta. The floor is yours. Thank you very much for having you around. Thank you.
Thank you. Good morning, everybody. Thank you to Dr. Aminu Chitu and the rest of the team at AfriScience Network. I feel very privileged to be given this opportunity to share just a little bit, a tip about microorganisms. So without much ado, I will just dive right in and start the lecture. I will share my slides now. Okay, so can anyone let me know if my slides are visible?
Yeah.
Yes,
they are. Yeah, thank you.
Okay, yeah, they are. Yes, they are visible.
Okay. All right, thank you. So before I start, let me just say that the topic is a very broad one. So I'll try very hard to do justice to it within the available time allocated to me. So perhaps after the lecture, we could have a little bit of maybe questions in case there are anything that are not very clear. So my topic this morning is the sustainable applications of microorganisms in the fields of medicine, agriculture, and the environment. Now, the reason why I chose this topic is because there's this idea, even amongst life scientists, that many microorganisms, all they do is cause harm and havoc to human beings and the rest of the world. But the truth is only a very, very little percentage of microorganisms are actually harmful. We have a lot of microorganisms that we can apply in different aspects of our lives. And microorganisms are very, very intelligent. So it's left to us as human beings to harness this potential of microorganisms and use them to our advantage. So, as an outline, sorry. So sorry. Let me just go back a little bit. I will start sharing it again. So sorry. I had a little glitch. Sorry, just a few seconds. I'll start sharing my slides again. Okay.
Excuse me, Al Hassan Zahraddin Muhammad, could you please switch off your video? You are distracting people. Sorry, please. Al Hassan Zahraddin.
The host should be able to do that. The host should be able to switch off everybody's video or mic.
Dr. Watsani, you should be able to do that on your side, please.
The host should be able to do it because your participants are not responding and they may have challenges.
Yeah, thank you.
Okay, are the slides visible now?
Not yet.
Okay, are they visible now?
No.
No.
So on my end, it's showing that it's visible.
Okay, yeah, they are visible now.
Okay. What you are seeing, is it the outline of the presentation?
I think it's one of
No.
the slides.
One of the slide, I think slide number 28. That's what I'm seeing here. Slide number 8 says role of microorganisms in the Sustainable Development Goals, SDGs. That is slide number 28.
Yeah, you have to go back to the beginning, ma.
Yeah.
I have gone back to the beginning on my end, so I'm wondering why it hasn't on your end. Oh,
maybe it is freezing, something.
It is now visible.
Yes.
The slide is now
Is
visible.
it outline of presentation now?
No, it's slide number 28.
Okay,
that is what is visible here.
Okay, let me stop sharing and try again.
Ma, can you just log out and come in again? Maybe it can be better.
Okay.
Sorry about that. It happens.
Yes. Okay, so let me go back. So do you want me to try sharing again? Are you still seeing the slides, or you want me to log out and log back in?
I think you can try to reshare it again. Let us see what will happen before you log out.
Okay. Should be coming up now.
Are you still sharing your slides? Because we can see something here. I mean, just an empty
Yes, I don't
PDF
know
file.
why it's, I think it's coming up
Okay.
now.
Yeah, I can see. Okay, fine. Now
Okay.
we can see it. But
Okay.
page 5 of
Okay.
33.
Okay, fine. Thank you so much. I'm so sorry for the
No problem.
delay. Okay. So I started by saying I will try to do justice as best I can within the time allotted to me. So this is a quick outline of the presentation. We'll look at a brief introduction about microorganisms and how they relate to the issue of sustainability. And then we'll look at the application of microorganisms in the field of medicine, agriculture, and in the environment, respectively. And then finally, we'll look at their role in the Sustainable Development Goals, because we can't talk about sustainability without talking about the Sustainable Development Goals. So to start off, what are microorganisms? So to a layman, simply put, microorganisms are those living organisms in which they cannot be seen by the naked eye except with the aid of a microscope. And that is because of their very, very tiny size. So you can find some of them being as small as micrometers, like the bacteria, and then some are even as small as nanometers, that's the viruses. So you have different kinds of microorganisms. You have bacteria, viruses, fungi. For the fungi, you have some that are actually very large, and then you have those ones that have to be viewed under the microscope, like the yeasts. Now, even the ones that are very large among the fungi, called the molds, they are filamentous fungi. However, you still will not be able to study their structure and morphology properly without looking at them under the microscope. And then you have some parasites. So parasites consist of things like helminths, which we call worms ordinarily, and protozoa. But these worms, their infective stages or the parts of the life cycle that are infective are usually very microscopic. So usually we group helminths and other parasites under microorganisms. So I mentioned earlier that microorganisms are very, very intelligent, and this is because they have been able to evolve over billions and billions of years. They have been able to cope with different kinds of climatic conditions, very extreme environments, and that has made them to be able to cope in any environment to find them. So as microbiologists, we know that microorganisms are ubiquitous. That means they are found everywhere. And the reason why you can find them everywhere is because they are able to adapt their metabolism to suit whatever environment they find themselves. That is why we have some of them living as normal flora within and outside the human body. You find them in soil environments, in water, in air, and even places where you have very extreme temperature, extreme salinity, you find microorganisms there. So that is why on this slide, it is mentioned that they are tiny, but they are powerful engineers of planetary life. So that word micro only describes their size. It does not describe their capabilities in any way. And that is because they are able to adapt to any kind of situation in which they find themselves. So just as a summary, microorganisms can be used in different aspects of sustainability. At the top of this chart, you have what is called bioremediation and detoxification of pollutants. So with the explosion of human growth and the increase in urban and industrial activities, we inevitably release a lot of pollutants in our environment, the soil, water, and even the atmosphere. So microorganisms are able to adapt to the presence of these pollutants and use them for their metabolic needs. And as a result of that, they're able to remove these pollutants from the environment and make it harmless to human beings. Microorganisms can also be used as biosensors, in which it can be used to sense the presence of certain pollutants in the environment or the presence of certain disease-causing agents in the human body. and thereby increase knowledge about the situation of interest. So microorganisms can also be used to fight plant diseases as a form of biopesticides. So the pesticides most of us are used to are the chemical pesticides. But if you use microorganisms as pesticides, you are able to kill the plant pests, and at the same time, you are not introducing any harmful substance to the environment. And also very, very important is the fact that microorganisms cycle our nutrients, particularly carbon and nitrogen, also other nutrients like phosphorus and sulfur. So you have different forms of elements that are not, should I say, reachable by living organisms, not just man, but plants. But microorganisms are able to convert different forms of different elements from one form to another. A very good example is the case of nitrogen. Now, if you take, for example, in the atmosphere, we have about 78% nitrogen in the atmosphere. But nitrogen is very important in the soil. Now, it takes a microorganism, a nitrogen-fixing bacterium, to fix gaseous nitrogen into a form that is usable by plants. So they fix atmospheric nitrogen to solid form of nitrogen, the form of ammonium and ammonia. And that way, plants are able to harness the nitrogen and use it as a form of nutrient, and the soil becomes fertile, and so on, the food chain can continue. Also, when it comes to carbon, we have a lot of debris and dead and decomposing plants and animals. It takes decomposers, which are usually fungi and bacteria in the soil, to break down this carbon trapped inside dead living organisms to a form that is now usable by other elements in the ecosystem. So we'll look at the first application of microorganisms. When we're looking at the sustainable applications of microorganisms in the field of medicine, our primary concern is health. How do microorganisms improve our health? How do we use them to prevent diseases and also to fight diseases? The first application is biocontrol. So from the name, biocontrol means you are using a biological agent, in this case, a microorganism, to control a pathogen. And a pathogen is a disease-causing organism. So the primarily means is the production of antibiotics by soil microorganisms, particularly those microorganisms that live around the root area of plants. So the root area of plants is called a rhizosphere, and the rhizosphere is a very rich environment because you have a lot of interaction between the plant's roots and the soil, and that's because you also have several organic acids and other beneficial nutrients around that area. So it also increases the amount of microorganisms that grow there. Now, many of these microorganisms that grow around the root area are able to produce antibiotics. So the first antibiotic that was discovered was actually produced by a fungus called Penicillium notatum, and it was discovered around World War II, I think. So when they had a lot of cases with bullet wounds and what have you, the antibiotic they used, the first antibiotic that was discovered was penicillin, and it got its name from the fungus Penicillium from which it was produced. So what happened is Penicillium species, particularly Penicillium notatum, produces a substance when it's in the lag phase or when it's not in the active phase of growth, which can kill bacteria. To be able to use particular antibiotics to treat the wounds. And thereafter, they were able to discover so many antibiotics produced by microorganisms. And you also have a large number of bacteria that produce antibiotics as well, particularly those ones that belong to the group of bacteria called the actinobacteria. And then you have some other bioactive compounds which are produced by bacteria, particularly lactic acid bacteria. So from the name lactic acid bacteria, it tells you something. But lactic acid bacteria actually produce the organic acid called lactic acid. So organic acids are friendlier and milder than your regular mineral acids, which we use in our chemistry labs, like sulfuric acid, hydrochloric acid. And because of their weak strength, they are able to live or dwell within the human system and then have a kind of benefit. So for instance, when you have lactic acid bacteria in the human gut, it controls the growth of several pathogens. Also, lactic acid bacteria produce substances that naturally prevent the growth of pathogens that will constantly come in contact with, particularly those ones that cause enteric infections or infections that affect our gastrointestinal tract. And they're able to prevent these pathogens by producing substances like peptides, bacteriocins, and some exopolysaccharides. That is why it's always encouraged to have a very good diet that has probiotic. Now, speaking of probiotics are actually microorganisms. For instance, Lactobacillus rhamnosus and other Lactobacillus species. However, you also have a fungus, which is a yeast called Saccharomyces boulardii, which also produces probiotic. So probiotic substances are very important to maintain our gut health. And when the gut health is maintained and improved, you find that there are so many conditions that are kept under control. For instance, our metabolism, our metabolic rates, which indirectly also affects weight loss. And it also affects inflammatory diseases like the BID, bioinflammatory disease, and other diseases which come up when you have an unhealthy balance of normal flora in the GIT. So the probiotics help to ensure you have a good number of healthy bacteria in the gut. Now, when it comes to medicine, microorganisms have also played a very, very important in the prevention of disease. And that means production of vaccines. So vaccines are actually produced from the target microorganism itself. So either a killed version or an attenuated version of that particular microorganism is used to produce the vaccine. And that is because when our body faces an antigen or a pathogen, it produces soldiers, let me put it that way, or white blood cells or antibodies. So these antibodies can now fight the disease and the person doesn't come down with the disease. So when you have vaccines, those vaccines, what they do is they elicit a reaction from the body as if the actual pathogen is in the body, thereby the body produces antibodies. So a lot of vaccines are actually prophylactic. So they prevent the disease before it comes in, and they are produced using microorganisms. However, we'll look at that in the table. You have a few vaccines that are actually therapeutic, in which they are used after the disease starts developing. Then you also have biopolymers, which are very, very important substances produced by primarily bacteria. So biopolymers can be used as a means of drug delivery, delivering drugs. So microorganisms, when they're under certain stress conditions, produce certain compounds, which can be extracted and used as a kind of bioplastic. So these bioplastics can be used to deliver drugs, anti-cancer drugs, and system because they are environmentally friendly and they are biodegradable and they do not elicit a negative reaction or tissue rejection when used in the human system. Also, microorganisms can be used to detect the presence of certain disease-causing agents in the body. So we look at some examples of the applications of microorganisms in the field of medicine. So on this table, what we have are different antibiotics. And then in the second column, you have the microorganism that produces this antibiotic. In the second column, what you have mostly are bacteria, but you also have some fungi in there as well. And then you have the kind of activity or their target organisms. So many antibiotics have a particular organism in which they target. For instance, the first kind of antibiotics, which are the penicillin, they attack what we call the gram-positive bacteria. However, you have some antibiotics that are broad spectrum. For instance, neomycin, it can be used against different kinds of bacteria and also other kinds of microorganisms. And then on this table, we'll look at different kinds of vaccines, the kind of pathogen in which they are used against, or the disease in which they are used to prevent, and then the kind of antigen in which result from using those vaccines. So you have the poliomyelitis disease, diarrhea. Diarrhea can be caused by different microorganisms, but in this case, we're referring to the viral diarrhea, which is caused by rotavirus. And then you have mumps, chickenpox, yellow fever, tuberculosis, typhoid fever. So if you look at the last column, you have P. So that P is telling you that they are used in the case of prevention. But if you look further down on the last row, you have T. So that T is therapeutic. So this particular vaccine is not used to prevent a disease. It's actually used to cure a disease. And that particular disease is not infectious. So infectious diseases are caused by microorganisms. So all the diseases that are caused by microorganisms, the vaccines that are given to the patient is actually to prevent the disease. But when it comes to something like cancer, this particular case of cancer is not infectious. You have some that are affected or caused by microorganisms, but this particular one is not caused by microorganisms. Prostate cancer, the vaccine given is actually therapeutic in origin. And then you have biosensors, which can be used in different capacities, not just in the field of medicine. So it can be used to discover several drugs and used also to detect diseases. And this is because biosensors produce certain reactions that generate signals that tell you pathogen or disease of interest is present in that place. And that is the same way it's being used in the environment when it's trying to detect the presence of certain pollutants. Now we'll look at the application of microorganisms in the environment. So the applications of microorganisms in the environment is very, very broad. And this is because most of the time you find microorganisms within the environment. So they have been able to adapt, unlike in the clinical setting where most of the time you have a pristine environment. So in the environment, you have more microorganisms, and that means you even have more potential for the capacity of usage. So here we have just a summary of some of the applications of microorganisms within the environment. The first one is in the field of bioremediation. So for bioremediation, if you look at that word, if you split it in two, you have bio and remediation. So bio coming from living organisms and remediation coming from to remedy or to provide a solution. So it just simply means you are using a microorganism to provide a solution. So from the environment. So like I mentioned earlier, because of our increase in industrial activities, we have so many pollutants in our environment. And sometimes the method we use in removing these pollutants may end up even becoming a problem itself. However, if you go green, you go the way of nature and use things like microorganisms or plants, you find that you are able to remove those pathogens successfully, and at the same time, you are not introducing another pollutants in the environment. So microorganisms, particularly fungi and bacteria, have been used to remove things like petroleum hydrocarbons in places where you have petroleum spillage or where you have leakage from petroleum tankers. They have been used to remove heavy metals from places where you have mining activity or places where you have heavy usage of chemical substances. You usually find heavy metals places where you use a lot of industrial substances. And then you can use microorganisms to remove things like pesticides. Now you find a case where we abuse the use of pesticides in the agricultural setting. So when we use too much of these pesticides, they become a problem and a serious pollutant in the soil. And eventually they can get washed into our water bodies and find their way to our system. So you can use microorganisms as well to remove pesticides and other organic pollutants that are coming from maybe the laundry sector, coming from where you have a lot of soldering activities, tanning activities, any kind of industry. Those pollutants can usually be removed by microorganisms, especially or unless rather they have that particular gene. When they have the gene that allows them to produce the enzyme that can enable them to remove these pollutants, they can be used for bioremediation. Also, in the field of petroleum, you can use microorganisms to recover petroleum from the soil. So that process is called microbial enhanced oil recovery. Usually they use a physical method, chemical methods, but there's a certain stage in which you can actually use microorganisms to help recover petroleum hydrocarbons from within the geological part of the earth. And then you can produce biofuels as alternative energy using microorganisms, particularly if you're trying to solve the problem of waste pollution. So where you have municipal waste, it could be agricultural waste, it could be residential waste, and then you have microorganisms that conduct a process called bioconversion, and they convert these waste substances to useful fuel. For instance, biogas, they produce, or what we call biomethane. They produce methane, which you can use for heating, lighting in our homes, and then things like bioethanol and biodiesel, which are very, very important now that we're trying to produce electric cars and we're trying to reduce our carbon footprint. So microorganisms can actually do a lot of waste to wealth conversion when it comes to handling waste in the environment. And the biopolymers I mentioned earlier in the case of applying microorganisms in the environment, in medicine, can also be used as packaging material because they are seen as bioplastics. So even if that's that aspect of producing biopolymers has not been fully developed because there are some challenges about plastic. The major challenge is that the bioplastic produced by microorganisms are not as, they don't have that tensile strength as the other petroleum-based plastics, but it has a lot of advantages because unlike the plastic, the regular plastics we know, the bioplastics are biodegradable, and that solves a huge, huge problem and is a huge win for the environment because plastics pollution has become a very big headache globally. So microorganisms, particularly those that bacterial species called the bacillus species, are able to produce substances, acid, polyhydroxyalkanoates, which can be extracted from the cell and then used to produce bioplastics. And these bioplastics can even be reinforced with organic substances, which ordinarily we may toss away, things like rice husk and what have you, just to increase their tensile strength, and they can be used as packaging material instead of relying on the petroleum-based packaging. Microorganisms, particularly bacteria, they can be used in bio. So that means they can be used to extract certain metals, for instance, gold, chromium. So biomining is a process in which you use microorganisms, particularly bacteria, to extract metal ore from the earth. So there are several countries that are already using biomining commercially. For instance, countries like Chile, South Africa, they use biomining in extracting their metal ore. And then when it comes to the field or the industry of mining, there is a lot of waste that comes up during the process of mining. So microorganisms can still be used to recover these metals from the environment, so you don't end up becoming a pollutant. So microorganisms can be used in metal recovery. And those particular kind of bacteria that do that are bacteria that are chemolithotrophs. So because they can actually live an environment where you have these metals and use them in the metabolic processes. They are able to take them out of the environment. Then you can use microorganisms in the production of bioconcrete in the construction industry. So that means you have concretes that have self-healing properties because there are certain bacteria that are able to produce what is called calcium calcites. And when they produce this substance, they're able to heal or seal cracks within concrete building materials. And then you can use microorganisms, particularly bacteria and some fungi, in prevention of corrosion in metallic substances used in the petroleum industry. I mentioned the petroleum industry because the petroleum industry is unique in the case that we have extraction of oil and refining of oil, and all this process enhances contacts between this liquid and surfaces of metal, and then you have a very slippery surface in which microorganisms can attach and start causing corrosion. So when microorganisms cause corrosion, you could also prevent this corrosion by using microorganisms again, because these microorganisms that prevent corrosion will prevent the growth of those ones that will cause corrosion, or they'll even produce substances that will allow corrosion to take place by the other harmful bacteria. So here we're just trying to explain in detail what bioremediation means. So simply defined, bioremediation refers to the use of microorganisms to break down or degrade contaminants that pose environmental and human risks. So the means by which microorganisms are able to break down, for instance, petroleum hydrocarbon, is that they produce enzymes that are able to cleave the hydrocarbon structure, either the ring, open up the ring, or break down certain bonds, and thereby they're able to use it for their metabolic processes and produce metabolites that can still go back into the regular system. And then they also produce what is called biosurfactants. So because petroleum or crude oil is an oily substance. When you have production of biosurfactant, what it does, it acts like a detergent. So what it does is it pulls the oily fragment and the watery fragment together. So when it pulls them together, the microorganism will now be able to break down the oil. So microorganisms are able to break down petroleum because of the production of certain enzymes which are specifically targeting hydrocarbons, and or, because not all of them produce biosurfactants, the production of biosurfactants. This chart is just simply telling us what happens during the process of biodegradation. So if you have your biodegradable substance, it could be a hydrocarbon, or it could be a pesticide. But in this instance, we're looking at a petroleum, kind of petroleum hydrocarbon. It could be an alkane, or it could be a benzene. So that hydrocarbon is an organic molecule. But the microorganism also needs nutrients to grow. So there are certain inorganic nutrients which should be provided to the microorganism within that setup. And then it also needs to have optimum growth conditions. So microorganisms have different preferences. You have those ones that can survive with little oxygen. You have those ones that prefer a lot of oxygen. And then you have some that don't even want any kind of oxygen. And also when it comes to temperature, you have those ones that can survive around the temperature that human beings also survive, the mesophiles. You have those that prefer very high temperatures, the thermophiles. And you have those that like the very cold temperature, like the psychrophiles. So depending on the microorganism you are using for biodegradation, you have to provide the optimum conditions which it requires to grow. And then it can break down that particular hydrocarbon or pollutant to simple substances. So that is why biodegradation and bioremediation is a sustainable solution, because it produces byproducts that are relatively harmless to the environment. So for instance, hydrocarbons are broken down to CO2 and water, and all the other metabolic byproducts are things that are in very small quantities and do not affect the environment negatively. And then we look at how microorganisms extract petroleum or oil, how they are being used. So in a regular setup where you have an oil rig, so this is like a kind of representation of what you have there. To the left, you have an injection well. And if you look from the left to the right, you have different colors. So those different colors are showing different stages of the oil recovery process. So when you have the bacteria of interest that can recover oil pumped into the well through the injection well, as also the bacteria and the nutrients which will sustain the bacteria pumped into the injection well, you have a horizontal kind of movement. So the first stage is the microorganisms start producing substances that allow it to thrive in that high oil environment and allow it to also break down the oil. So these substances include biosurfactants, biogenic acids, some enzymes, solvents. So all these things allow the microorganism to break down the oil or crude oil in the core of the earth. And then it moves from that stage to another stage in which you have the wettability of the oil is affected, is altered, so that it can now be extracted from the rocks. I think they are sedimentary rocks. And then you have breakdown of those substances from where they are usually found, because petroleum are actually gotten from dead organic matter, dead plants and animals. So these substances produced by the microorganisms allow them to be able to extract the oil from the sedimentary rocks where they are usually found, and then eventually through the production well, the resulting crude oil is pumped out of the earth crust. Now, in the mining industry, like I mentioned earlier, microorganisms, particularly sulfur bacteria, are used in biomining, and they can also be used to restore mining sites or acid mine drainage sites, because they can be used to remove those ones that also be pollutants. So there are two ways in which microorganisms, two stages actually, in which microorganisms are able to remove these metals. The first one is they fix the heavy metals biologically, and then they form these heavy metals once it gets into their cell. And the reason why they're able to do this is because those particular bacteria have some anion groups in their cell membrane. So the cell membrane is the lining just immediately after the cell wall. The cell wall is outside the cell, and then the membrane is within the cell. So when it does that, it's able to absorb the cations coming from the heavy metals. So the anions are membrane of the bacteria, and then what comes in most heavy metals are cations, so they have positively charged group. So when the heavy metal positively charged group comes into the cell, it attaches or fixes to the anionic or negatively charged groups within the cell of the bacteria, and that way the bacteria is able to precipitate or remove the heavy metal from that particular environment. So here we're looking at the circular bioeconomy and how different factors and different mechanisms in which microorganisms are sustainable to the environment, how they all interplay and how they all come together. So when you have recovery, energy recovery and composting, particularly where you have waste, you have the production of bioenergy, alternative energy sources, biofuel, bioethanol, biogas, and what have you. And this energy can come into the economy and be used as a source of revenue and a source of improving the lifestyle of the citizens in that country. So when you have a lot of waste, those wastes can be channeled to use by microorganisms, and the byproducts coming from the utilization of this waste can also be used as bio-based products. For instance, you could have biosolids, biofertilizers that can still be used in the farms to fertilize the soil, and they can also be used as fuel in biorefineries. So it helps the economy of any country if there's a lot of investment on the use of beneficial microorganisms, because it has a triple beneficial effect. So when it comes to bioconcretes, on this slide, you have two different photographs. The one on the left is showing you how there's healing taking place coming from the calcites, the compound produced by the microorganism. So most of the time, you have bacteria like Leuconostoc species, Bacillus species, using this substance. So if you look at the photograph on the left, you find that at the top of the photograph, you have a yellowish substance. That is the calcite produced by the microorganism. And that is what glues or cements the crack within a concrete. Then if you look on the right, the other photograph is showing the process where a microorganism is applied to a particular concrete with a crack, and how over time the size of the crack has reduced, and eventually you have the crack completely closed. And this is because of the application of bacterial cells of interest that produce the calcite compound that heals the concrete and make sure the crack stays closed. So this is from a study by Alemu and others in 2022. So the biopolymers, which can be used to produce bioplastics, the two most common ones are the PLA and PHA. You also have PHB, but PHB is a kind of PHA. So they are very useful in producing bioplastics. There have been a lot of studies in using them to produce biodegradable plastics, and they have a lot of advantages over the petroleum-based plastics, because those ones are very, very difficult to degrade. Over time, it takes a long period of time, like hundreds and thousands of years to be broken down, unlike the bioplastics that are biodegradable by microorganisms. And these particular biopolymers are more acceptable by the human body. So that is why it is being used as a means of drug delivery. So able to achieve greater success in transplants and other kinds of surgeries. So the next photograph, we're looking at a site where you have corrosion, and that corrosion is actually caused by microorganisms in the petroleum industry. So, but there are three ways in which microorganisms in the petroleum industry can prevent or inhibit corrosion by other microorganisms. The first thing is that they could prevent the growth of those ones that can cause corrosion. Now, those ones that cause corrosion actually produce things that can cause digestion of the particular substance. For instance, acids. They produce a lot of acids. So when you use microorganisms like the ones I've mentioned on this slide, when you use them on those sites, you prevent the growth of those ones that can cause corrosion. Also, another way in which they prevent corrosion is that they remove those corrosive substances, for instance, the acids that allow the metal surface to be corroded. And then some of these microorganisms, another mechanism they use is that they can form a layer by covering the surface of that metal that is susceptible to corrosion by other microorganisms, thereby preventing the growth of the corrosive microorganisms. Now, how do we use microorganisms in the field of agriculture? There are many ways in which we can use microorganisms. And the primary reason is because the soil is the richest place where you can find microorganisms because of the kind of nutrients you have there. So among the three components of the environment, soil, water, and air, soil is the one in which you have natural microorganisms. But for water and air, they only find themselves there through man's activities. But for soil, you actually have microorganisms that are called indigenous soil microorganisms. And that is because the soil is very rich. And because the soil is referred to as the sink of the earth, that is where everything comes in. There are a lot of nutrients and other compounds that get into the soil and that aid and promote the growth of microorganisms. So from this photograph, you can see the caption there. There are more microorganisms in a handful of healthy soil than there are people on earth. Actually, even in 1 teaspoon, you have more bacteria. Let's not even say microorganisms, more bacteria than people on earth. And that is because of how vibrant the soil environment is. So just to summarize the kind of applications you can use microorganisms in the field of agriculture, you have, as they can be used to produce biopesticides, biofertilizers, and then they are important in nutrient cycling and carbon sequestration in the soil. Then they can prevent diseases from plant diseases from occurring, and they can also promote plant health by promoting shoots, plant roots, and microorganisms in the soil can also be used to form what is called biocrust, in which the integrity of the soil is maintained and corrosion is reduced. So we'll look at each of them briefly. So in the field of agriculture, microorganisms can be used primarily for plant growth and development. But how do they achieve this? Through many ways. One of the ways is through nutrient cycling. So nitrogen fixing, phosphorus solubilization, they allow a lot of nutrients. That is why most of the fertilizers and nutrients we use in our soil are made up of nitrogen and phosphorus, because they are the two most important nutrients when it comes to soil health. So you have microorganisms that are important in fixing nitrogen and converting nitrogen from one to another form. And then you have microorganisms that also solubilize phosphate salts that are found in the soil. And in so doing, the soil is now able to extract the portion of the phosphorus that is important for plant growth. Then you also have microorganisms that produce substances that fight or kill plant pathogens, called biopesticides. So in so doing, it allows the plant to grow healthily and to develop and run its normal course. And then you have, for the case of carbon cycling, you have the decomposition of organic matter. And microorganisms, particularly fungi, are the primary drivers of carbon decomposition. And then you have other bacteria coming in place, particularly those polysaccharides and cellulolytic substances you have in the soil. And then finally, you have microorganisms that are able to support different kinds of stress, biotic stress and abiotic stress in the soil. So those are ways in which microorganisms aid plant growth and development in the soil. So when it comes to biofertilizers, from the name, it just means a means of fertilizing or increasing soil fertility using microorganisms. Now, there are several ways, but we'll just focus on these three. The primary one is nitrogen fixation. And even the nitrogen fixation, you have different kinds of microorganisms that fix nitrogen different ways. You have the symbiotic nitrogen fixers. Those ones are the ones that have a symbiotic relationship with the plant roots, particularly leguminous plant roots. So that is why in a process like crop rotation, you find that you have legumes being planted after a period of time. And that is because farmers have noticed over time, they have noticed that when you plant protein, the soil is richer and more fertile. And the reason is because leguminous plant roots have what is called root nodules. So those root nodules, you have nitrogen fixing bacteria present in those root nodules, and they have a symbiotic relationship, and then you have nitrogen fixed in the soil, which makes the soil more fertile and richer. And then you have those bacteria that live, free living bacteria in the soil without forming any symbiotic relationship with the plant roots, yet they are able to fix nitrogen in the soil. And then you have cyanobacteria, which are also called the blue-green algae. They are also important in fixing nitrogen in the soil. Then you have the phosphorus solubilizing bacteria. So those ones increase the availability of phosphorus to plants, and that is because those phosphate salts are tightly bound in the soil. They're able to produce acids to ensure that they become soluble, and when they are soluble, they are now easily accessible to plant roots, and thereby increasing soil fertility. For example, are the bacillus species. And then you have what is called the phosphate mobilizing bacteria. So those ones, phosphate mobilizing microorganisms. So those ones primarily are the mycorrhizal fungi. So mycorrhiza has a combination of two words. So rhizae has to do with the roots, and myco has to do with the use of fungi. So those are fungi that are living in plant roots. You have those living inside the plant roots, which are called avuscular mycorrhizae. And then you have those living outside the root, which are called ectomycorrhizae. So when you have mycorrhizae in soil, it increases the integrity of the soil because it improves the soil structure. So you have less washing away or leaching of nutrients from the soil, thereby the nutrients are being retained in the soil. So when you have this symbiotic or mutualistic relationship between this particular fungi and the plant root, it increases the retention of nutrients in the soil, thereby making the soil more fertile for plant growth. Now, when you have nitrogen fixing bacteria in soil, the first photograph you are seeing, where you can see below the soil surface, you see a lot of roots and some colored substances. So those colored substances are actually microorganisms, particularly the actinobacteria that you find around the rhizosphere. So those microorganisms are usually the ones that have symbiotic relationship with plant roots, and they have symbiotic nitrogen fixation. So the process of nitrogen fixation just involves trapping atmospheric nitrogen, so nitrogen in the form of gas, and converting it to solid or liquid form, which now becomes available to the soil. And then you have the pesticides. So the pesticides are substances that are naturally produced by living organisms like plants, microorganisms, and animals. But most biopesticides, particularly the one that is being known globally, that's the BT, is produced by a microorganism called Bacillus thuringiensis. So BT is being used commercially in many countries, including the United States. So BT is a safe way of controlling plant pests in agriculture because it doesn't have the negative side effects you find with chemical pesticides. So it is a very safe way, very sustainable way of treating plant pests in the field of agriculture. So this particular microorganism produces genes or certain substances that has the ability to kill the pests. So these genes can be extracted and can now be introduced into the plant so that as the plant is growing, the pest is not able to attack or harm the plants. So use of biopesticides is environmentally friendly, but each biopesticide has a particular host, so it is host specific. In this photograph, we're just looking at an example of how the BT is being used in the field of agriculture. So for here, you have the European corn borer, which is the plant pest. So you can see it looking very happy with its cutlery about to feed on the maize plant. So, but at the top, you have Bacillus thuringiensis, which is the bacterium that produces the pesticide. So the name of the gene that allows it to produce that enzyme or that protein that is harmful to the plant is called BT. So that BT gene is extracted from the bacterium and inserted into the maize plant so that the pest, once the pest has contact with the maize plant, the BT gets into the system of the pest and kills the pest. So the pest is not able to harm the maize plants. So in the field of agriculture, you have what is called biocross. So biocross is a combination of different microorganisms which you find in the soil. And usually they are important in dry lands or desert areas, because that is where you have a problem of very little nutrients in the soil, because you have less water, you have more sunlight, less moisture. So you have very, very high cases of not enough nutrients in the soil. However, if you have the application of biocross on the surface of the soil, you are able to ensure that you have nutrients in the soil and plants can actually grow in the soil. So biocross ensure that you have less loss or removal of water and nutrients, and also the effect of soil erosion is combated, so you can have retainment of nutrients in the soil. In this photograph is just an example of what the surface of a soil that has received biocross treatment, what it looks like. So this is from the desert in the Grand Canyon in the United States. So the Grand Canyon is a very, it's a desert area. So this is a sample of one of the research activities where they have applied biocross to the surface of the soil to see the effect it will have overall. So how are microorganisms important in the SDGs? So on this slide, you have a summary of about 7 different research areas. So even though in this talk, I mentioned just medicine, environment, medicine, environment, and what's the third one again? So you have also the use of microorganisms in food and industry. So that one is a very, 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 very long application. However, have a lot of food industries in which microorganisms are being used as taste enhancers to make pastries, for instance, bread, to produce things like yogurt, dairy products, alcoholic beverages, non-alcoholic beverages, for fermentation processes. So there are so many applications for food, and microorganisms can be used to combat or to ensure that you have food security. So that is one way. Another way is in the prevention of disease, which we have looked at, prevention of human disease and even plant disease. So microorganisms have a very good potential in improving the health of a population. You also have the production of alternative energy or clean energy, as opposed to fossil energy using fossil fuel, using microorganisms. Because microorganisms can be used to produce bioethanol, biodiesel, biomethane. And you can use microorganisms to ensure that the biogeochemical cycles are maintained, because microorganisms are the ones that ensure that you are able to convert one form of an element to another form, particularly is actually required. So other ways in which microorganisms are important when you talk about the SDGs is in recycling. So particularly when you talk about waste conversion. So you can use microorganisms as a means of recycling, particularly when we talk about bioplastics and talking about conversion of waste products to renewable form of energy. And then also in the use of microorganisms to remove pollutants from the environment, particularly bioremediation. And finally, in restoring the ecosystem. There are many ways in which microorganisms can be used to restore the ecosystem, either from by way of removing industrial pollutants or by restoring polluted sites using microorganisms. So there are 7 major ways in which microbial technologies can be used to achieve the Sustainable Development Goals. But speaking of the SDGs, the particular SDGs in which microorganisms can be used are about 11. So you have SDG 2 when you talk about zero hunger, because microorganisms can be used in food security, good health and well-being. We have seen applications of microorganisms in the field of medicine. Clean water and sanitation, microorganisms can be used for wastewater treatment, as biosensor, and so many applications when it comes to clean water. You can also use microorganisms for clean energy, affordable, clean, renewable energy. You can use microorganisms when it talks about decent work and economic growth, because when you have healthier lifestyles, they are able to work better. You can use microorganisms for different innovations and infrastructure in the industry. You can use microorganisms in the field of sustainable cities, because microorganisms can be used in the built environment, not just for bioconcrete, but also in ensuring that certain regulations and policies allow our living areas to be safe. When we go back to the COVID pandemic, we see that the way you design your structures and your indoor environment can affect the spread of airborne diseases. So studying microorganisms can ensure you have sustainable cities. You can use microorganisms for responsible consumption and production, for climate action, also for life below water and life on land. So microorganisms have a lot of potentials when it comes to their sustainable development goals. It's left to us to dig into this potential and harness them. The problem we usually find in a country like Nigeria is actually lack of funding, but we don't lack the ideas to be able to promote all these ideas. And then if you look at the Sustainable Development Goals and how it is all linked to the applications of microorganisms and the tools in which are necessary. So if we want to start using microorganisms, like so many countries, for instance, have started commercializing the use of bacteria in cleaning up oil spillage, particularly in a state like Texas, where they have a lot of petroleum activities, they have so many commercial products of microorganisms that they can use to clean up oil spillage and what have you. And that is because they have the right environment is there, because you have enough funding, you have so many sources of funding, not just from the government. You have the necessary technology, tools, training and retraining to learn all these skills to be able to get the microorganisms to that stage, because you can't use a raw microorganism like that in the environment. You have to go through a certain process before you can use it in the environment. So if we don't have the right environment in the country and the right tools and technologies, this knowledge will just be wasting without us being able to harness them. So if you look at this chart, it just talks about how all these tools and the use of microorganisms and the applications are interconnected, and then the different fields in which you can use them. So when you have bioprospecting, so in bioprospecting, you do it like you are prospecting for petroleum products. So in this case, you are bioprospecting for those microorganisms of interest, those ones that are beneficial, those ones that have the gene that allow them to conduct those applications or processes in which you are interested in. When you have them, you have to conduct a lot of research to ensure that you have the right mix of microorganisms. You have to ensure that it has the necessary metabolic tools. You have to ensure it has the right shelf life, because you want something that wouldn't die out. You also want something that will not become pathogenic with time. And then you also need to have the necessary computing skills to analyze these biological data, particularly bioinformatics, to analyze the gene, the genome, and then looking at so many other things that has to do with the gene of that microorganism. And then you now move to the stage of development. You now move to the R&D. How can I upscale this property of this particular microorganism that I can use it for large scale, and it eventually helps my nation, and then it helps me to be able to target and achieve certain SDG goals, SDGs. And then you have, from that stage, you move to commercialization. That is where the circular bioeconomy comes in play. That is where you can see if you are actually reaping benefit from using these microorganisms. And then finally, you have your end product. It could be in the use of microorganisms in the industries. It could be in the agricultural sector. It could be in the petroleum industry, and what have you. So these are ways in which they are all interconnected. So in the field of synthetic biology, you have several innovations, and microorganisms have a big role to play in the advancement of synthetic biology, particularly in Nigeria, in which some of these technologies was still just coming up with these skills. So microorganisms, even if they have this ability, you can actually increase the likelihood of these abilities and capabilities being used. And how do you do that? By manipulating the gene so that you have more production of that particular substance. And then you also increase the ability of that microorganism to withstand abiotic stress so that it can be more useful for longer period of time. So some of the sustainable innovations include using them to treat wastewater, to break down plastics eventually, and then clean up a lot of pollutants, because heavy metals are pollutants you find in almost every environment, not just in the industrial environment, but also from the residential. And then you can also use them to produce renewable energy. So like what we have mentioned before, biodiesel, bioethanol, biomethane. You can also use them to replace chemical fertilizers and pesticides, which we use in our farms, in our agricultural sector. So you now have, instead of using those chemicals, you now start using biopesticides, biofertilizers, and then you can have those microorganisms, not just using them in that capacity, but fitted with biosensors so they can be able to have a higher impact or produce a much better outcome when they have been used. So in conclusion, I would like to say that even though we have a lot of microorganisms that are beneficial, there are also some limitations and challenges. Sometimes when you work with microorganisms in the lab, upscaling them to apply them in the field, in the environment, in the field of medicine, you may encounter some challenges, and that is because they are living organisms. And sometimes living organisms can be unpredictable, but there are all these limitations can be bypassed with a lot of technological advancements. So you have to study these microorganisms properly and not just studying them as a lab scientist or biologist, but also using other tools, using transcriptomics, omics technologies, and bioinformatics to understand how you can get the best of these microorganisms. And because we're in Nigeria, I will also say that a very big problem is funding. So you may have beautiful ideas, but if you don't have the necessary funding for that idea, it's very difficult for you to upscale it from one point to another point. Another problem, again, has to do with our bureaucracy in the country. So we don't have an environment where you are allowed to tinker and play with these microorganisms as you deem fit in the environment. So a way in which we can combat this is if we have a lot of industry and academia collaboration, and then you can find that so many of these beautiful ideas can come to fruition. So we're not saying none of them have been used. We have so many of them that have been used, but I think we can have them used at a much better scale and at a larger capacity. So I would like to end my talk today by reminding us all that microorganisms may be invisible invaders, but they are amazing allies as well. Just have to make them our friends and understand them and know how to harness them. So with this, I would say thank you very much to the AfriScience Network. A big thank you to my mentors and professors and my collaborators. So I welcome your questions. Thank you.
Thank you very much, Dr. Habiba, for this insightful webinar talk. Honestly, the topic is very interesting. It reminds me of my time when I was in the secondary school, science secondary school. This really, yeah, all those things in biology and agricultural sciences, and this shows the just the applications of those ideas. Personally, I like applied sciences, and that is what some of us are doing nowadays. All those things in mathematics, and we are trying to see how we can use them in studying the behavior of infectious diseases. So this is actually very interesting, and personally, I'm excited, and I believe I'm not the only one. And before we go on to the question and answer session, I don't know, should I hold on? I have a question, honestly. Should that be the last? Okay, I think I have like two in one question, but I think in your conclusion remark, you made mention that it is on the applications of all these technologies that you have mentioned, small scale applications. Some of them are at experimental level, like where you made mention of the insertion of BT component in plant, etc. So that's more or less like a kind of experimental stage, or is it something that at a developmental stage, not only this insertion of BT component in plants, and is it something that is at a developmental stage, something that is only achievable the lab or it is into practice. I mean, in a large or commercial, especially in developed countries, as you mentioned that some of the limitations here in developing countries, Nigeria in particular.
Okay.
And then,
so, but I think I had just one question. Like, is it at the developmental? Okay.
the second that I have say the ground. Epidemiology of infectious diseases. Yeah, that's what, yeah. So like a 2-in-1 question I have.
Okay. So, are you done now?
Yes, I think so. You can go ahead and answer the question.
Okay. I don't know if it's from my end or his end, but it kept breaking up. Did you experience the same thing?
Yes.
Okay. Aha. Okay.
Yes.
So, but I know he asked if it's just at the developmental stage. So, for the issue of BT, for treating and preventing plant crops that are being grown commercially, because you have large area of land, so they have usage of BT. So it's not just at developmental stage, but for Nigeria and some other developing countries, we may have not started using it. I'm a master student, but it was against the tomato Ebola. We used same BT and we're able to get very good results, very, very good results. But unfortunately, we're not able to move from that stage to commercializing it. So that's where we stopped. But I don't know, we have very sound agricultural institute. I don't know if now they have started commercializing BT and using it in mass. So, but for other countries outside Nigeria, in the developed world, yes, it's being used. It's not at the developmental stage. Unfortunately, I didn't hear the latter part of his question, so I'm not able to provide answers to them, except maybe if he sends it by.
Dr. Shitu, are you back? Okay, I think he's not back yet, but we can go ahead and take other questions within the remaining time that is left. Mabel, can you ask the question? Please unmute yourself.
Good morning.
Hello, Mabel. Good morning.
Yes, good morning, Dr. Habiba. I'm Dr. Mabel Idika from University of Ibadan. I see Dr. Habiba is smiling. Do you remember me?
You look very familiar. I'm trying to place the face.
Okay. It's such a really small world. I came across this notice on the UI WhatsApp platform, and I was so shocked. Like, I know this prof. I know her. I'm sorry. I don't have a I don't have a question. I just want to say that was a good presentation, and we can catch up later. I'll remind you from Kaduna.
Thank
We
you very
go
much,
back way back, yes.
Yes. Mabel, if you don't mind, if you're in the group chat of the AfriScience Network, maybe we can connect there.
No, I'm not.
Yeah, there.
No, I'm not. I said I just came across this notice and I decided to join so that I can reconnect with you.
Oh. Okay, please send me your phone number in the chat.
Okay, I'll do that.
Thank you.
Good to see you again.
Same here.
Yes, Professor Fashina, you can ask a question, please.
Yeah, thank you very much. And thank you to Professor Atta for this presentation. It's a very good presentation. Indeed, it relates to what we've been discussing recently on AfriScience Forum, on how the industry need to connect with the academia. You know, the issue of town and gown, we've talked so much about it, but we hardly put it into use in Nigeria in particular. I've seen a number of countries, because I've worked across a number of African countries, and I've seen them putting this to use. And we've shared just as late as early this morning, we were sharing our experience on the forum on this type of issue. The microbial ecosystem offers us a lot as scientists, both in terms of making money available to the university, creating opportunity. For example, if I can give a quick example, in the University of Nairobi, I know in the Faculty of Agricultural Science there, I know they produce yogurt. And they use all this microbial system to produce their yogurt. And I've been there one time and they were complaining of fund. And I asked them, why don't you then scale up this yogurt and make it on a commercial scale? And as we speak now, a partner was at the university just about a week ago, some partner from the West. And they were talking to them about scaling up this productivity. I think it would be important for us to begin to catalyze some of these ideas, not just to talk about them theoretically, but how to begin to either, I don't know who we need to talk to in the Nigerian system, whether the NUC, whether the Federal Ministry of Education, whether Ministry of Science and Technology, and begin to bring out critical issues that come out from discussions like this. Indeed, we've had a number of these, both on our forum as well as on this type of presentation. Bring out those critical issues and at least send it across to the necessary agencies that need to hear about this. Some of the funding needs that we are talking about in the university system, we can even bypass it by issues like this. In the universities in South Africa, they make a lot of money. I worked there for a number of years, and we make a lot of money without even thinking of the government. Indeed, I know of a year wherein the government relied on the university, took some loan from the university in some of the years when I was in South Africa. So there are ways by which we can begin to convert the opportunities around us. And the microbial ecosystem offers us a lot of those opportunities. So we shouldn't see the microbes as negative agents alone. They offer us a lot of benefits, and we should take advantage of those benefits that we've seen. And this is also to speak to my sister that has recently spoken. So we invite you to also join us on AfriScience Forum. You will meet a lot more people than Professor Atta alone. And we are waiting for you. We hope that you will join immediately as soon as we finish this program. So thank you very much. Bye.
Thank you.
Thank you very much, Prof. Thank you.
Thank you for the invitation.
Thank you very much.
Well, I think the thing is, Yeah, I was just going to say briefly that the thing is, I think we have some of those things in place, but it's like we're not making the most of them. Like there are so many initiatives. Maybe we don't have enough because of our population, and then maybe we are focusing on things, on other things, and we're not looking towards microorganisms. Because I remember several years ago when I went to supervise some private students in NNPC, and I found my students in a section of the refinery where she was not relevant, you know. And I asked her supervisor, I'm like, what is she doing here? She's supposed to be in the HSE department or something like that. And he said, you know, that it's as if there's a gap somewhere. So if I even tell an engineer, okay, we can work together, we can talk about corrosion in the petroleum industry or oil recovery or bioremediation, you know, it looks strange to them. So I think maybe the machinery is in place, but there's a gap somewhere. So I don't know. So I think it still has to do with what you're saying, the gown and the sound. That is the issue. We need to find that there are ways in which we can collaborate and work together. That's just it.
Thank you very much, ma'am.
Thank you. Oh, sorry, please. I lost a connection at some point. Okay, Dr. Shoitan, you have a question. Professor Shoitan?
Okay. Thank you very much. Let me first commend Dr. Abila for your interesting and illuminating presentation. It's quite illuminating, and for bringing the applied and special aspects of microbiology. Congratulations again, ma. Just to collaborate what the last said, I've also been to University of Nairobi a couple of times, and I'm happy to hear that they are doing something about milk production. University of Nairobi has a large and viable cattle production, and when you get there, you start wondering that if this university can tap into this, they can start making lots of money. So I want to appeal to the last speaker, if he's hearing me, the points you raised, if you can type them in words and send it to one of the platforms in AfriScience, which you can share around. As Dr. Shiku mentioned, AfriScience is going places, and some of the discussions or points we made, they have a way of getting to the stakeholders and the policymakers. And I'm sure something will be done about it. Because on our platforms, we have vice chancellors of universities, we have director generals of research institutes, and many big and accomplished people, which I'm sure they read our comments. Some of them might not post or respond, but I'm sure they read our comments, and some of them send private chats to us on suggestion. So if the last speaker is online, please, if you could kindly type or summarize your comments and share it on the AfriScience platform that you belong, so that we can share it around. These are one of the little ways in which we can contribute to the development of our institution, because we keep hearing funding problem. Whereas there are some things that if we look inward, we have some resources that with little assistance, it will help the universities to be sustainable. Thank you very much.
Thank you,
Yeah.
Professor
Thank you, Prof.
Shoitan. I think, yeah. Sunday Habila, yeah, you have been raising your hand for some time.
Yes, thank you very much for the acknowledgement, because I was about to nag because my hands have been hot. Thank you very much for the opportunity to speak. And Professor Atta is my colleague here, and I didn't expect any less of this anyway. And again, she had been my classmate from the primary school. We attended the same primary school in Kaduna.
Yeah.
So I didn't expect any less of this exposition that I've had also. Now, looking at the exposition that we've had, the tripod of the presentation on the application of these microorganisms that we are talking about. You know, I am an urbanist, you know, with special interest in urban and environmental management and urban services and sanitation services as well. I got interested in the aspect where she was connecting these applications with the issue of SDG. And I'm happy SDG 6 was mentioned and SDG 11 was mentioned. However, I got interested in the aspect of water sanitation and hygiene. And we try to see how we can get access to clean water, because that to a large extent will affect our personal hygiene and sanitation. However, just like the saying, water everywhere, but there's no water to drink. How can we use these microorganisms, not just in wastewater treatment, but the water treatment for public water supply is my concern, especially in the aspect where we have heavy metals being eroded or flowed or moved into the water reservoirs. The last time we had some talks about the watershed of Ahmadu Bello University, sometimes last 2 weeks, and there are a lot of threats due to the problem of inappropriate and undesignated management of solid waste. And these things are being washed into the water reservoir of Ahmadu Bello University. And we do not have the technology to detect these heavy metals. So I am just wondering, are there microorganisms that can be used to take care of this? Because I see the actions of the microorganisms being more of life-to-life kind of intervention, life intervention. That is, if you have allowed me to say, organic to organic intervention, which I think will be very, very viable. I just want to know, from your perspective, Prof, are there some special microorganisms that can be introduced? Because by your presentation, I'm meant to understand that the microbes, are naturally in the soil and also the atmosphere. But in the water, they just find themselves there, introduced there by activities of man. So how can this be enhanced? Because my fear is the endemic, consequence of these heavy metals on our population in terms of planetary health, public health, which is also a concern, my concern, because I'm a registered environmental health officer. And anytime I hear this, the first thing that comes to my mind is the public. So how can this be helped? How can we use this to help our situation? Thank you very much.
Okay, thank you so much.
Yeah, Dr. Habiba, I think can you take the last question now so that you can answer all of them at once?
Okay.
Okay, so, I think I will ask Dr. Aguda Temitope Olusheye to respond because it's past 11:30 already. Yeah. Okay.
Thank you so much. I'm a technologist in King's University, Odeomu, Osun State. And I'm also a PhD student in Ladoke Akintola University of Technology. I first of all want to appreciate the good presentation from Dr. Habiba. I just want to ask for the privilege if young researchers like us can be part of an AfriScience network. If we have the privilege to do that, we appreciate if maybe the link of where we can get to be part of the group, such that we can be more enlightened on things like this, particularly in the application of microorganisms. And secondly, where I will stop because of the time, is application of microorganisms in nanotechnology. I saw the last picture showing an application around nanotechnology from microorganisms. Biosynthesis of nanotechnology from microorganisms have been proven to be biocompatible, but despite the biocompatibility, it is still contestable in the field of science. I don't know if Doctor can speak to this, Ma. Thank you.
Okay, thank you very much. Yeah, Dr. Habiba.
Okay. So that means I'm responding to the two of them now, right?
Yes, please. Yeah.
Okay. So thank you very much for the kind words and the questions. Very interesting questions. And big thank you to my my childhood friend, Dr. Habiba. So the last speaker asked a question which is somehow related to Dr. Habila's question regarding use of microorganisms in waste treatment. So a very good way of, actually, let me put it this way. Microorganisms have two roles in wastewater treatment. The first role is they can be used as indicators to tell if there are the presence of other pathogens, because you have a huge number of microorganisms in water that can cause waterborne diseases. But you have a few microorganisms that are used as standard indicators. So the first thing is microorganisms can be used as indicators of the presence of pathogens. Then the second one that's related also to the last speaker's question is the fact that you can use nanotechnology, so microorganisms on nanoparticles, to treat wastewater. And the interesting thing is not just heavy metals. So chemical pollutants, biological pollutants can be removed using these nanoparticles. And yes, there's a lot of compatibility. And thankfully, there's been a lot of advances in the use of nanotechnology in wastewater treatment. So now you have more pollutants being removed from water. The question now is, do these facilities have the resources to do that? So to answer the last person's question, yes, you can use nanotechnology and to a much wider scale than it was previously used. The only question is, that particular facility, do they have the resources to be able to apply this nanotechnology? And it's completely safe. Thank you. I hope
Hello,
I've answered all the questions.
Yeah, am
Yes,
I audible, please? Okay, for the last question on how to join the AfriScience network, I'm having a network problem here. I'm on mobile phone. I have posted it on the admins group. I want one of them to share it before we end this session. Okay, it has been delivered on the admin platform. So, and I would like to invite Dr. Ismail Odetokun to give us the closing remarks.
Yeah, thank you very much. First and foremost, I want to appreciate Dr. Habiba Atta for the great presentation you have delivered today. We are highly honored to have you within us and to give this talk. And I'm very sure that all of us have learned one or two things, and we are ready to take the challenge further than what you have presented. I want to say that it is a blessing for us to have somebody like you within the country, particularly at the Ahmadu Bello University. So I want to encourage young scientists to look at the caliber of Dr. Habiba Atta and ensure that they give their best to the system. Yeah, I want to state further that the AfriScience network is open to give more invite more speakers to give lectures in the nearest future. Every month we have our webinars coming up, so we want to encourage us to stay tuned and join the lectures whenever these adverts are being made. And I also want to encourage us to invite other scientists that are not with us now, that they should try and join the network. They stand to gain a lot in terms of networking, connection, and so many other things. So I want to appreciate members of the admin, particularly Dr. Aminu Shitu, Professor Fashina, Professor Shueton, Dr. Yabo, Dr. Wazani, Dr. Mary. So we wish every one of us a happy weekend ahead. Thank you very much.
Thank you very much. Yeah, thank you very much. Yeah, thanks, Dr. Habiba and all the team members of the AfriScience Network for the wonderful work we are doing in bringing African scientists together, share ideas, collaborate, and we are happy that we thank all the members, honestly, because their support, honestly, is what is keeping the AfriScience Network alive, and we are realizing our aims. They're actually working. So most of the things that we are sharing, they are working, especially on educational opportunities. Some of the members have succeeded in gaining positions abroad for study, either for postdoc jobs and PhDs, postgraduate studies, and a lot of questions related to study design, data analysis have been answered for students that are stuck, including myself. I was once stuck. I asked a question and it was answered very correctly, and I got it solved. So thanks to everyone. Thanks to members for their support. Thank you so much. Thank you very much.
Thank you. Too.
Thank you.
Bye.
Thank you.
Thank you very much.
Thank you, sir. Thank you so much.
Thank you, everyone.
Thank you,
Thank
everyone.
you. Dr. Habiba.
Yes, Dr. Shitu.
Yeah, if you can share with me your slides so that I can share them on the AfriScience network.
Okay, I'll do that now. Thank you once again for having me.
Thank you so much. We should be the one to thank you, honestly, because you just make our weekends. Yeah.
That's good
Thank
to
you
know.
very much.
Thank
These
you.
are
At
the
least,
contributions that keep us alive, honestly.
yes. All right. Bye-bye. I'll be in touch.
Okay, all right. Thank you. Take care.
Thank you. All right. Bye-bye.
Bye.